In this chapter we cover major concepts in tooth development. The first big concept is the timing. So far everything has been measured in weeks, but in this chapter we measure time in weeks, months, and years. Both the primary and succedaneous teeth begin developing early in embryonic development around week 5. To put this in perspective, that is only shortly after the mouth forms the oropharyngeal membrane ruptures in week 4. The process of tooth development begins around week 5. The next big concept is the importance of placode, or induction. Small regions of epithelial cells in the oral mucosa are induced to speed up mitosis. This happens because of secretion of morphogens from neural crest cells below. The third big concept is the lineage of the different parts of a tooth and periodontium. Enamel is produced by cells derived from the ectoderm, whereas dentin, cementum, pulp, PDL and alveolar bone are derived from neuromesenchyme. The fourth big concept is that the mesenchyme is derived from mesoderm, but tooth connective tissues are derived from neural crest cells plus mesoderm. We give this mesenchyme a unique name called neuromesenchyme. The fifth big concept is that enamel and dentin-producing cells do not form separately. They induce one another in a reciprocal fashion. After their team effort, the cells move apart from one another and do their own thing. The sixth and final big concept is that the process of tooth eruption is complicated. It is important to learn about the development of the PDL, alveolar bone, and junctional epithelium. Development of tooth. Tooth formation, or odontogenesis, starts with primary teeth around weeks 5 to 6, and succedaneous teeth around weeks 9 to 10. Teeth arise from invaginations of epithelium, derived from ectoderm. At week 5, the oral cavity is lined by ectoderm-derived epithelium, and deep to that is mesenchyme. Teeth, however, grow from ridges called the dental lamina, found on the mandibular arch and the maxillary processes, along the occlusal border. Here, mesenchyme contains clusters of neural crest cells that migrated from the neural tube. By 8 weeks there are 10 tooth buds in each dental lamina. The tooth buds of succedaneous teeth begin forming around this time, before the primary buds have made any enamel or dentin. Both primary and permanent buds develop into crowns and become surrounded by the maxilla and mandible. Induction. Neuromesenchymal stem cells coordinate with one another through planar cell polarity signals to induce the correct number of tooth buds, at the correct distance apart from one another. Neuromesenchymal stem cells secrete a number of morphogens forming complex gradients. Overlying ectoderm responds to the morphogen gradients. Two important morphogens in tooth development are members of the FGF and the BMP families of signaling molecules. At this time and location, they induce ectoderm to differentiate into tooth buds instead of oral mucosa. One of the first visible responses in the ectoderm is proliferation. Bud stage. Tooth buds begin to appear around week 6, and ultimately 10 tooth buds form on the maxillary processes and mandibular arch. In addition to proliferation of ectodermal cells, neuromesenchymal stem cells also proliferate. This makes the mesenchyme in a tooth bud appear denser than the mesenchyme in nearby regions that form the lamina propria of the oral mucosa. Around week 10, the ectoderm continues to proliferate and it bumps into the cluster of neuromesenchymal stem cells below. This cluster of neuromesenchymal stem cells is called the dental papilla. The dental papilla forces the ectoderm of the tooth bud to grow around it. As this happens, the ectoderm resembles a hat, hence the name cap stage. At this time, the dental lamina has two prominent parts, an inner and an outer layer. The epithelial cells near the dental papilla are referred to as the enamel organ, because some of them differentiate into cells that produce enamel. Below the dental lamina, the neuromesenchyme of the dental papilla forms dentin and pulp. The neuromesenchyme on the outside of the dental lamina is called the dental sac, or dental follicle, which forms cementum, PDL and alveolar bone. All three together, the enamel organ, dental papilla, and dental sac, are collectively called tooth germ. By the 11th or 12th week, ectoderm and neuromesenchyme continue to proliferate. As the epithelial cap becomes larger, it resembles more of a bell shape, hence the name bell stage. At this time, the enamel organ contains two layers of cuboidal cells named the inner enamel epithelium, IE, and the outer enamel epithelium, OEE. The IEE is next to the dental papilla, with the basement membrane physically separating the two. The IEE differentiates into ameloblasts and produce enamel, while the OEE is involved in tooth eruption and forms the junctional epithelium that bridges the tooth surface and oral mucosa. Past the stratum intermedium are ectodermal cells called stellate reticulum, named for their star-shaped rather than cuboidal appearance. These cells help induce the IE to differentiate and begin enamel production. These cells are found in the superficial but not the deeper regions of the growing tooth germ, which is why enamel is only produced in crowns and not in roots of teeth. The dental papilla may be subdivided at this time into an outer region and a central region. The outer cells differentiate into odontoblasts, while the central cells form many cell types found in pulp. Animated representation of odontoblast and ameloblast differentiation, including the induction signals, red arrows, for each process, during the bell and crown stages of tooth development.